Welcome. In this session, I'm going to discuss the last part of biological classification, that will be viroids, prions, and lichens. So let's begin this session with the viroids. So these viroids are first discovered by T.O. Diner in 1971. So they are first discovered by T.O. Diner in 1971. And these viroids, they consist of only RNA. So it consists of only RNA. So there is no protein code like how we see in case of viruses. Okay, so no protein code. In case of viruses, nucleic acid is present and that is covered by protein code, but in case of viroids, there is no protein code, only nucleic acid present, and that too it has only RNA, and that RNA is single stranded circular RNA molecule is present, and this single stranded circular RNA molecule is of low molecular weight. So it has low molecular weight, and these are the pathogens. So viroids are pathogens and cause diseases only in plants. So they are pathogens and cause diseases only in case of plants. Like for example, the potato spindle tuber disease, potato spindle tuber disease. So this potato spindle tuber disease is caused by a viroid called potato spindle tuber viroid. So potato spindle tuber disease is caused by this potato spindle tuber viroid, and this tuber viroid, okay, as I told, all these viroids will be having a single stranded circular RNA. So this is also a single, okay, stranded RNA molecule. It has single stranded RNA molecule. So that's about viroids. The next one we'll see about prions. So the next one, prions. So here, these prions are proteinaceous infectious agents. So they are proteinaceous infectious agents, and these cause several neurological diseases in animals and humans. They cause several diseases. Okay, so those neurological diseases, okay, they cause neurological diseases, means concerned with the nervous system. So they mainly attack the nervous system. So they cause neurological diseases in animals and also humans. So neurological diseases in animals and humans. Okay, so here these prions are actually made up of only proteins. So made up of only protein, and that too it is a abnormally folded protein. So it is a abnormally folded protein. So a functional or a normal protein will have a particular, okay, way of folding, whereas in this case of prions it is abnormally folded. Okay, right. So let's say here this is a abnormally folded protein. So abnormally folded protein, and say here, so this is a normal folded protein. So this is protein normal protein. So when this abnormally folded protein comes in contact with the normal protein, okay, then it can result in this normal protein to abnormally fold in the same way. Okay, so if this one comes in contact, and this will also, okay, start to fold in the same way. Okay, so it becomes, so this normal protein is now changed to abnormal protein. Okay, so this abnormally folded proteins, they in simple called the PRP proteins. Okay, so they stand for the PRP, okay, proteins. PRP protein, that is abnormally folded protein. Next, coming to its size. Okay, so prions have similar size, okay, similar size to that of viruses, which means prions and viruses have similar size. So prions, I'll mention, so prions and viruses have similar size. Okay, and let's see the diseases caused by these prions in animals and humans. Okay, so I told you that these are infectious agents and they mostly affect the nervous system. So here, in case of animals, bovine spongiform encephalopathy. Okay, so in short, it's called BSE, is a disease. Okay, so this is commonly called mad cow disease. Is commonly called mad cow disease, and this is seen in cattle. Seen in cattle. Okay, then and another disease called Creutzfeldt-Jakob disease. So in short, it is called CJD. So this Creutzfeldt-Jakob disease is seen in case of humans. So in humans. Okay. So this CJD is a, okay, fatal degeneration, okay, of brain cells. Okay. So here there is a degeneration of brain cells. Okay, this results in the death of the person. So that is it's a degenerative brain disorder. Is a degenerative brain disorder. Okay, so that's about prions. And the next one, we'll see about lichens. Okay, so already we know that lichens, so here is a symbiotic association. So symbiotic association between, okay, alga and a fungus. Is a symbiotic association between an alga and a fungus. So here a symbiotic association, which means two different species living together in which both species will get benefit. Okay, so here these are two different species living together, and okay, the alga gets benefit from the fungus and the fungus gets benefit from alga. Okay, so this algal component of a lichen, so it is called phycobiont. It's called phycobiont, whereas the fungal component is called mycobiont. So phycobiont and mycobiont. Okay, so this phycobiont is autotrophic. So it is autotrophic, which means it is capable of synthesizing its own food materials. So I will say it is photo autotrophic. So that means it will perform photosynthesis, performs photosynthesis. Whereas the microbiont component is heterotrophic. Heterotrophic. It will not synthesize its own food materials, nor it performs photosynthesis. And what it does, the microbiome, so this microbiome will provide shelter. So it provides a shelter to the alga, and then it helps absorbing mineral nutrients. Okay, so it helps in mineral nutrients and water, so which it will supply it to the alga. So by taking these minerals, water, the alga will perform the photosynthesis, and in the photosynthesis, it synthesizes food. So it synthesizes food. Okay, some of this food will be used by the alga, and some food is provided to fungus. Okay, so some of this is provided to fungus, next so these lichens are good pollution indicators. So lichens are good pollution indicators. So here the meaning of good pollution indicators is that they do not grow in polluted areas. So do not grow in polluted areas. Okay, especially if the areas are having sulfur pollution, and so they will be not growing in those polluted areas. Okay, and here these lichens are of different types basing on their shape. So we will see some three types of these lichens basing on their shape. Okay, so basing on shape, so the lichens are of crustose type. So crustose lichens. Then foliose lichens, and then fruticose lichens. So crustose, foliose, and fruticose lichens. So here crustose, which means encrusting. So they are uh, present, growing flat on the substratum. So crustose. So they are appressed to the substratum. So crustose lichens. Then foliose, which means these are leaf-like. So leaf-like lichens, and then the fruticose, which will be the shrub-like lichens. Okay, so these are flat and closely attached to substratum. So they are flat and closely attached to substratum. And here coming to the folios, they are already I mentioned leaf-like, with lobed or irregular folded margins. Okay, so leaf-like or lobed with irregular folded margins, we will say. And then coming to this fructose type, so they are branched cylindrical. These are branched cylindrical and appear, okay, shrubby. So they will be appearing shrubby. So let's see some examples in this case, okay, about these crustose lichens. So crustose, for crustose lichens, an example is, okay, caloplaca. Okay, so caloplaca, right, flavescens is an example for this crustose type of lichens. Next one, foliose type. Okay, so for this foliose type, so it will be hypogymnia physodis. So hypogymnia physodis is an example. And then lastly, the fruticose type of lichens. So for this cladonia, okay, evansi is an example. So these are different okay, types of lichens based on their shape. Right, so with this, we complete this chapter, biological classification synopsis. In the next session, we'll see objective questions on this chapter.